Before I start, two questions. One, who in this room considers themselves to be more of a biologist? Okay, a few of you. And who considers themselves to be more of an engineer or a physicist or something else? Okay, so we're half and half. Well, darn. My apologies to the biologists in the room. I am not a biologist, but I'm really excited about engineering new tools for biology and for medicine. And what I'm going to tell you about today is a new technique that my lab has been working on, which I hope in the future will be able to solve some of the common problems that I saw articulated across the wonderful talks that we've had so far today. And I'll tell you about that in just a minute. So thank you so much to the organizers for inviting me. I'm absolutely honored to be here speaking to you all today. And yeah, all right, so let's go. So, good. So I'm going to tell you three things. The first thing I'm going to do is tell you about the challenge that we would like to address, where I see space for us to do something new and deliver a new tool that's useful. Then I need to tell you something about how we're going to make the measurements that I say we can make, and that's going to require a little diversion down instrumentation lane. And then finally, we'll come back and show you that these things that we've engineered do in fact work, and talk about some of the things that we can use them for, hopefully genomics in the future. Good. All right, so first, let's talk about the challenge. The challenge is multiplexing at the single molecule scale. So today you've seen lots of beautiful images of cells, and here's one that I stole from the internet, of beautiful imaging of, I believe, actin filaments, and let's see, that must be the ER, not the mitochondria, so one of the biologists has to help me here, and a nucleus, great, it's a cell. But we know that there's a lot more going on inside cells. So with apologies to David Goodsell, we've heard a lot today about DNA-protein interactions, protein-protein interactions, how all of these dynamic interactions at the single molecule level are governing the fates of cells, whether that means that they're senescent or cancerous, right? And okay, to study these, maybe you can, well, we like to take them out of their cells and study how they're interacting with one another. And to do that, fluorescence is a really useful tool. It helps us monitor their interactions. And separately, my lab has sort of another side that manipulates them. And we use fluorescence to see what they're up to, right? So inside or outside of a cell, fluorescence is a really useful tool. But what you'll notice here and in some of the talks that you saw earlier today is that you're seeing fluorescence in like 3 colors. So another question, who here has a microscope that shows you one color of fluorescence at least or more in their lab? Or your PI has one, that's okay, that counts. How about 2 colors? Anybody? Loud and proud. 3 colors? 4 colors? Ooh, all right, Northwestern crowd, good job. Anybody do more than 4 colors? Melika, I know you guys do multiplexing, but right, so to do more than 3 colors, you have to play some tricks, right? And that just happens because spectra are really broad. So what we want is to be able to label all of these biomolecules at once. Wouldn't it be nice if we could see where different genes are dynamically and what proteins they're interacting with in real time, and then watch what happens to the cell as a result? Now, okay, that's very blue sky. We cannot do that yet, but what I'm going to talk to you about today is hopefully a step down that path. Okay, so why is multiplexing hard? Well, with the color axis, with spectral multiplexing, we know that fluorescence signals, fluorescent emission, spectra tend to be quite broad and they overlap. Now, if you have maybe a 5-channel microscope, you want to get real fancy, this is fine as long as you're measuring big objects that have lots of fluorophores in them because you have great signal-to-noise ratio. So not only can you tell a red from a green object, but you can use the ratios between those channels to identify lots of different types of objects. And people have used this to great effect. For example, Brainbow to look at thousands of different colors of neurons or thousands of different colors of beads. These are all ratios of a few color channels because they have great signal-to-noise. But at the nanoscale, to follow one molecule, you get very few photons. Somebody earlier today said 500. We hope to get thousands from ours, but it's really just that, thousands. That means that your signal-to-noise kind of sucks. So ratiometric sensing, you can get maybe 10 ratios instead of 1,000. If you want to get really crazy, and I do mean really crazy, this is 4 alternating lasers with special timing, lots of different fluorophores. People have been able to show up to like 25. And so this is the problem that we're trying to solve. So at the single molecule level, one trick that you can use is find additional dimensions to separate that aren't color. Color is one great axis. Let's find other ones. This could be all sorts of things. People have used time. You can do multiple rounds of imaging with the same color fluorophore, but attach it to different antibodies or maybe to different pieces of DNA so that it sticks to different things. And you can identify them sequentially and see lots of different colors that way. That works great. Maybe you can spatially separate a few colors, and that sequence that you see uniquely shows you, oh, it's this construct and not the one that went blue, green, red, right? You can also use kinetics. DNA paint has at least been alluded to today. You can have, if you can tune the strength of your interactions, you can tune your on and off rates, then you can tell the difference between something that is binding very fast and leaving and something that stays there a long time. And so maybe they're the same color, but they can be different colors, right? So that's useful. Okay, but all of these approaches, even though they get you hundreds or even thousands of distinguishable objects or interactions, these require things to hold still, and biology is bad at holding still. In a cell, everything is moving around, or in a sample, maybe you have a diagnostic sample from a patient, you don't want to have to wait and fix things to a slide. You want to know right now what's in there and what's it doing. Okay. So here's your other option. Your other option is to find other axes of fluorescence that you can see in one shot. Those might be things like lifetime. Everybody here knows what fluorescence lifetime is. That's how long it takes for a photon to pop out of a molecule after the molecule's been excited. Maybe it's polarization. You know that the molecule is a dipole, and so it's going to emit photons with polarization preference, right, according to how it's oriented. So if you can orient it, maybe you can tell it apart from its buddy that's oriented differently. Now, with lifetime, for example, here I'm showing 3 of my favorite red fluorophores, and they all have very similar emission spectra. They are all red. So if I just look at color, they're very hard to tell apart. But this is a great example of how lifetime, how long it takes me to get photons out after exciting the molecule, allows me to tell them apart. The problem with this is that these 3 guys all have different chemistries, and I'm not a chemist, but when you take different chemistries and put them in a messy cell which has different pHs and salts depending on where you are in the cell, that means that these 3 molecules are going to behave differently. They're going to respond differently to different areas of the cell. So using different chemicals is kind of a problem for multiplexing, even though it does some of what we'd like to do. You can also use demixing. So let's say that you have a few different red dyes, a few different green dyes, and so on. You can just look at different areas of the cell and maybe tell apart, like this I think did maybe 9 different dyes. Jiao? 9? Okay, good. Okay, so all this to say, spectroscopic properties are useful for taking one glance at a molecule and figuring out that it's different than something that is technically the same color, right? But different chemistries are a problem. Okay. Bear with me. Okay, one more piece of info I want to tell you is that FRET is useful. FRET, probably most of you are familiar with. This is Forster resonance energy transfer. And all it means is that if I have 2 dipoles, 2 fluorophores that are close to each other within about 10 nanometers, they feel a resonance when they get excited, and they can transfer a virtual photon between them, usually from the higher energy fluorophore to the lower energy fluorophore, so that instead of just green fluorescence from my higher energy fluorophore, I might see red fluorescence because the energy gets transferred to the red fluorophore and then emitted as red. The closer they are, the more efficient FRET is, and the farther they are, the less FRET you see. So biologists like to use this, no offense guys, just to say, did stuff bind? Do I have a low FRET or do I have high FRET? Biophysicists like to get a little bit more specific and start thinking about the orientations and moving molecules very specifically, using this as a molecular ruler to tell us how far apart things are, not just whether or not they've bound. And in particular, I want to highlight that DNA is a great way to program how far apart fluorophores are. You can order a piece of DNA from IDT and order a complementary strand and know that your fluorophores are going to be 8 base pairs apart exactly. And if you do that, you can measure different signals, right? And this is kind of what we're trying to do, right? We're trying to create different signals that are differentiable. So FRET is useful, but FRET isn't quite enough. Here I'm showing you, this is an awesome paper, and they're seeing 15 different signals. And they placed these fluorophores on DNA to get all these different signals, and then they ran out of space because FRET stopped working because things got too far apart. So FRET isn't quite enough, even though it's a really good way to get us about an order of magnitude of multiplexing. Okay, last thing I need to tell you before I tell you what we actually did. This is, if you can tune something about the fluorophores properties in addition to changing FRET, then you can move the FRET curve around in your detection space. Here's what I mean. So here we've got a super simple computational model of DNA. It's just a double helix. Here's a green fluorophore. Here's a red fluorophore. So we can simulate what signals we should see in terms of red brightness and green brightness, the lifetime, the apparent lifetime of the green fluorophore, and what the FRET efficiency is. And the blue curve shows me what happens in my control simulation, where everything has the normal photophysical properties. If I then change, the red curve shows you what happens if I just decrease the quantum yield of this guy by just 15%, and the green curve shows you what happens if I change its lifetime by 20%. Both of those changes move that curve of FRET locations. One moves it in this brightness projection, and one moves it in this other lifetime FRET projection. So this tells us that if you're able to tune the photophysics of one or both fluorophores, it can move this whole curve of FRET objects around in your detection space. Okay, 3D view, it's wonderful. Great. Oh, and we know that this is possible, that you can make, I guess any photochemists in here are saying, but quantum yield is always coupled to lifetime and they should change together. We know that if you change the dielectric environment of a fluorophore, you can separately alter its quantum efficiency and its lifetime. Okay, that's the background you need. Now let me tell you what we did. So what we did is we created different FRET constructs, super simple. This is just on DNA, green fluorophore, red fluorophore, and we put the red fluorophore at different positions. This gives us 10 or 15 constructs that we should be able to tell apart. And then we changed stuff like the sequence or maybe the mechanical properties of the DNA right around the green fluorophore. So we changed the attachment chemistry so that here you can see it's kind of squished. We got rid of this base opposite it. Here we attached it just with one linker instead of with two. And here we stuck it on the end. These are really simple changes, but it turns out that these change the photophysics of that dye so that we can move our FRET curve around in our detection space. Everybody with me? Okay, so now let me tell you how we're actually going to read out all this complicated photophysics so that it can actually be useful. We use something called an ABEL trap. An ABEL trap is an anti-Brownian trap. Everybody's familiar with Brownian motion. Maybe you're looking with a confocal microscope at this one spot in solution, and if you're looking at little fluorophores, they like to diffuse in and out of that region. And so you get these random bursts of photons as things diffuse in and out. Even those random bursts carry a lot of information. But wouldn't it be nice if your molecule just stayed right there so that you could measure it for as long as you want until it photobleaches? That's what we do. We keep track of where an object is relative to where we want it to be, which is right in the center of our trap. And if it tries to move at all, we smack it back into the middle with voltages. We're using a little microfluidic cell. We've got little electrodes stuck into it, and that's kind of it. Well, the computer's doing all the hard parts. We do this every single time we see even one photon. And when you turn on feedback, so here things diffuse, now we turn on feedback, and now we have a molecule stuck in the center of the trap for as long as it will fluoresce. And this means that our signal looks way better. We get way more photons, and it's a much cleaner signal than you would get with these very sensitive detectors on something moving randomly. Okay, so for the microscope geeks in the room, just really quickly, we're sitting on top of an inverted microscope. We have an excitation source that is pulsed and polarized so that we can measure all this fun stuff. And we scan it across the center of the microfluidic cell in kind of a grid. That's so that we know where the molecule is. We control all this with an FPGA, which does really precise timing, so that every time we see, again, even one photon, the computer can estimate where the molecule must be and apply an appropriate voltage to kick it back towards the center, just by electrophoresis, or, okay, if you have a neutral molecule, electroosmosis. Happy to talk about that later. While you have the molecule trapped, you can do whatever you want with all the rest of the photons. You can split them up and do polarization. You can split into color channels. You can take a spectrum on an EMCCD camera. The world is your oyster. You can do your own photon budgeting. Okay. Good. The kind of data that we see from this are photon by photon arrivals in different polarization and color channels. You can add them together to see the brightness of a molecule over time. You can take a ratio to see its polarization, its anisotropy as a function of time. You can look at their lifetime decays. So every photon that comes in has a delay from the pulse of light that excited the fluorophore. You can build up histograms of those to show you the characteristic lifetime decays for your object. And you can look at the spectrum. This is actually a calibration spectrum of a Heine laser, but you can see how the spectrum maybe changes for different states of the molecule. Okay, so just an example of this guy in action. Oh, actually, this is important. And, wait, there's more. Because we know the record of locations where we saw the molecule while we were trying to trap it, and because we know the pattern of voltages that we applied to counteract its diffusion, we can back calculate what its free diffusion coefficient would have been if it were allowed to diffuse. That means that we can tell the difference between a big molecule and a small molecule. And we also know something about its mobility. How well did it respond to the electric fields that we applied? This is going to be useful later, I promise. Okay, so we can tell lots of stuff. If you take that away from the slide, I'll be happy. Okay, let me just show you an example that is totally not related to genomics, but is another side of what my lab does. Here we've got a light harvesting system. It's got tons of fluorophores in it. They're all doing FRET. Energy is moving to a specific spot. When we throw it in the ABEL trap, you can see simultaneously we read out brightness, polarization, lifetime, and the spectrum, or spectral center of mass. And we can accumulate these points onto histograms. Here I'm showing you, hopefully, different projections. Brightness is always on Y, polarization, lifetime, and spectral center of mass, and the spectrum over here. And for this sample, which is a control, everything accumulates into one photophysical state, one population. Clustering. Great. Lots of points, one cluster. This is good because this is a control. If you look at something that's a little bit more photophysically complicated, for example, this guy plus another little accessory protein, don't worry about the biology, we can even see from the single molecule traces that stuff now looks weird. The spectrum's jumping all over the place, the lifetime's jumping all over the place. Maybe they're correlated, not clear. Again, the biology doesn't matter, but when we accumulate enough points, what I want to show you is that we can very precisely see these two different photophysical states that are very close to each other. And in this case, this showed us, to the biologist's surprise, that there were a couple of these guys binding and that they were binding at specific locations. So this is just to show that we can differentiate small differences among photophysical states. Okay. End of sidebar about instrumentation. Back to multiplexing. So I told you that we made these objects, we move the Psi5 around to change FRET, and we change the attachment chemistry on the Psi3 so that we can shift the curve. And now what we want to know is, how many of these can we see? Can we label biomolecules with them? And can we then tell those biomolecules apart correctly, which hopefully we would be able to. Let me show you that we can do that. We sense these in the ABEL trap. We dump a bunch of them into our microfluidic cell. They get trapped one at a time. And you can see this is a mixture here. They have different signals. The reds and green channels show different ratios. Each molecule that enters has a different FRET ratio. Great, good start. They also show us different lifetimes. This one here, green lifetime is about a nanosecond. And we see in a different part of the trace where the red guy has photobleached that the green lifetime is longer. Great, so far so good. Okay, so we wanted to prove to ourselves that indeed the different chemical changes that we make near the donor are changing the lifetime, so we checked what that did. You'll be able to see that, don't worry too much about the details, but basically this population is going to move around in our detection space depending on what our attachment chemistry is. And I'm leaving out some details here, but you can see we are, by changing the attachment chemistry, we can change where that population shows up. Yeah, and that's all that's saying about that. Okay, so that works, great. So our lifetime and our quantum yield are tuned, and they are tuned independently by the ways in which we are changing these chemistries around the molecule. Okay, so how many of these things can we see? We made a few different sets of molecules. Here's one set with this attachment chemistry, red fluorophore in different places, different attachment chemistry, red fluorophore in different places, and so on. And what you can see is that indeed these populations that we see fall in different areas of our detection space. On the top row, you're seeing projections into red and green brightness space. On the bottom, you're seeing projections into green lifetime and fret space, which there are similar patterns in both, but it's useful to be able to see both views because some things will overlap in one view but not in the other. Okay, so we can see lots of these. We made 40-something of these different constructs, and we verified that indeed they do all fall in different areas of our detection space. So the next challenge is figuring out what set of them we can use together. Some of those blobs I just showed you do fall on top of each other. You can do, Camilla just showed this, a confusion matrix to figure out, if the real thing I have is AB7, what's the probability that I can fuse it with any of these other constructs that I've made? The ones that are confusing, we chose a threshold of 2.5% probability of getting it wrong. If it can be confused, you can't use that pair together. And eventually we picked this set with the arrows on them to use together in one mix. We also looked at how many photons we need to make a reasonably confident assignment, like, oh yeah, that's fret floor number 23. And it turns out, so here I'm showing, these are all for what's called AB11, this one construct, and it has close neighbors, AB10 and AB12. And when you compare it to AB10 and AB12, you need a lot of photons in order to get your ID correct. And by a lot, I mean a few thousand, about 5,000 or 6,000. If you have something that's further away, AB16 is more different, okay? Very easy to tell it apart from AB16. So this is to say, you could pick from this little palette of labels that we give you an appropriate set for whatever your application is so that your identification of molecules is maximally orthogonalized. Okay. This is just for the data geeks in the room. This is some raw data coming out of the ABEL Trap. So we got red and green channels, and the blue, if you can see it, shows lifetimes of all these levels. Every time the brightness goes up, we have a new thing in the trap. So we can take average values of these states. We can get the fret levels. We can eliminate the things that I've made dim are the ones where maybe one of the fluorophores is photo-bleached or something. We screen those right out. Everything else we can assign to being one of this mixture of 27 different fret floors that we threw all in one pot to tell apart. So it works pretty well. And if I accumulate all this data and a bunch more with it, then I get these scatter plots here. Again, this is showing red and green brightness projection, and then lifetime fret projections. And I've had to slice this into two pieces because the brighter slice falls on top of the dimmer slice when you look at it from above. And actually, I think it's easier to see in 3D. The point is, we took 27 of these tags, we threw them all in one pot at very low concentration. This is tens of femtomolar each. And you can tell them apart. These are pretty tight clusters. We can confidently call the identity of each one. But these are just the constructs we've made. Now we want to actually use these things as labels. Tens of femtomolar. Yay. Good. Okay. Well, it's DNA. You can stick DNA to things, right? It's really easy to, all things considered, it's pretty easy, okay, to stick DNA to something else that's a nucleic acid. So for example, you can design this little bridge that's complementary to a loop, a predicted loop on mRNA, and then your little DNA tag will stick, sequence specifically to that mRNA. Or you can have it invade the end of a double-stranded DNA. Or you can use your favorite attachment chemistry to stick it onto a protein. And what we see is that, remember how I told you that in our trap we can tell the difference between a small object and a big object because we can measure diffusion coefficient? That means that if I mix together these fret floors with mRNA, some of the fret floors will stick to mRNA and others won't. The ones that don't have higher diffusion coefficients. They move faster in the trap than the ones that stuck to an mRNA. That means that you don't have to wash your samples. For someone who is not a biologist and does not enjoy purification, I really like the idea of being able to throw things together and then make a measurement instead of going through steps to purify. Oh, and the signal doesn't change. Great. Okay, and so finally, we detected mixtures of these biomarkers, again, tens of femtomolar concentration. Here's a couple different mRNAs together where we had one extra fret floor in the mix just to make sure we didn't have any off-target binding, and indeed, we don't. We can see the EGFP mRNA and this luciferase mRNA. The orange and the red are the ones where the fret floor found its target. All the points in gray behind are fret floors that were moving too fast to have bound an mRNA. And then we tried a bunch of other stuff in the same pot with it. And indeed, we can see double-stranded DNAs and free fret floors and proteins that are labeled with fret floors. And they form these nice little tight clusters. Anything in a color has been confidently identified as being that fret floor, that object. Anything, one of these like real light gray dots, those are dots that we could not assign. Okay, so this is just a baby step towards something that's going to be useful for genomics. But what we're envisioning next is using this on the one hand for imaging. Wouldn't you love to be able to see all of your favorite genes and transcription factors at the same time, or maybe super resolved, who knows, in a cell, in a living cell, right? Or you could use it for diagnostics. You have a biomarker panel for a cancer, and you want to be able to take a teeny amount of sample, maybe even just from your little spheroid, your tumor model, and figure out what cytokines are in there, what biomarkers are in there. So we're thinking about all of these applications for next steps. Good, so today I've shown you that we can make and tell apart these fret floors. We've made them from 3 simple components. This is actually, I think, the most important thing about this approach is that we managed to generate lots of distinguishable signals from chemically similar things, so that if you go into different areas of the cell, they all behave kind of the same way. We see them right now in an ABEL trap. We're working on doing that with more traditional imaging techniques now. And we can detect very low abundance mixtures of proteins and nucleic acids, which we think potentially has some exciting applications. Right, so with that, I want to briefly advertise both Jiachang and Kavya are here today and giving posters. Arya, a postdoc in our group, is also here, but not giving a poster. Jiachang will answer all of your questions about this multiplexing approach. Kavya's been working on this a bit too, and she's our ABEL Trap expert. She's got all kinds of new bells and whistles and applications to condensates in biology. So yeah, definitely go harass them at their posters when you get a chance. And I just want to thank my wonderful group, especially the folks who worked on the project that I've talked about today. Ayesha and Jiachang are the co-first authors on this paper, which hopefully should be out like next week in Nature Nanotech, yay. And Kavya and Arya are both here today, and the rest of the group is wonderful. So many thanks to them, our funders and collaborators, and to all of you for paying attention at the end of a long day. Deeply appreciated. Thank you.